Welcome back. The last lecture, we left off with a problem. Many things in the environment may be causing cancer, and we have no way of evaluating which ones do and which ones don't. We need some better tests. I want to introduce this topic of cancer and things in the environment with the story of Ernst Winder. Ernst Winder was a medical student at Washington University in St. Louis, and in the late 1940s, and he attended an autopsy of someone who had died of lung cancer, this man. And the man had horrible black lungs, and Winder was very astounded at this, and he looked on the medical record, and he found that the man who was a lung cancer sufferer had been a long-term cigarette smoker of at least 2 packs a day. Now, we know that the link between smoking and cancer was over 200 years old. Remember the stories in epidemiology from Great Britain. But Winder wanted to do serious epidemiology now, and he did some case control and cohort studies. And when he did these studies, he found the relative risk for long-term smokers was well over 10. It was sometimes 15 to 20, depending on how long they smoked. The tobacco companies found out about this linkage between tobacco and the potential linkage between tobacco and cancer, and of course made a massive counterattack. They claimed that lung cancer is caused by air pollution, could be, by diet, that also could be. The families of smokers who had died sadly sued the tobacco companies. And they were given the runaround, and the tobacco companies had big lawyers, and they delayed and delayed, and they kept investigating and saying, no, it's not this. So what was needed was a smoking gun, for want of a better term. We needed something that would link something in tobacco smoke to cancer. And what do we mean by something that causes cancer? Of course, from the previous lecture, we mean damaging DNA. Now, tobacco smoke has over 3,500 chemicals in it. And at least 60 of them have been shown to cause cancer in lab tests and in animal tests. The challenge was to link a specific chemical in cancer to, a specific chemical in tobacco to human lung cancer. One of the chemicals that was a good suspect is benzpyrene. Benzpyrene, here's the molecule, is a flat molecule that is in cigarette smoke. And benzpyrene is kind of like aflatoxin. It binds to guanines in DNA and thereby causes mutations because the DNA replication machinery comes along, just as I described for aflatoxin, sees this, doesn't see a G, it sees a mess, and puts in any old base opposite it, typically an A, but other things. There is a gene that was focused on for these studies that people focused on, and the gene is called the P53 gene. P53 is a tumor suppressor. The protein product of P53 guards DNA from damage. It's called the guardian of the genome. It's one of these DNA things that is involved with DNA repair. So if P53 isn't working, you have a bad, and because of DNA mutation, there's going to be more damage to DNA because of mutation, and the damage will not be repaired. Turns out that P53 mutations are very common in cancer. The DNA gets damaged, and the growth is uncontrolled. And when you look at where on the DNA gene these mutations are, they seem to be clustered in certain locations. We'll describe the significance of this later on in the course. These locations are in hotspots. So it's kind of like saying, if I put my pen somewhere in the body, where am I going to put it? I'm going to put it in a pocket, right? The mutations for P53 on the DNA gene were at a certain location with regard to its expressed protein, and those locations were just at 1 or 2 places. So here's a map that shows you along the DNA genome here where the mutations were. And those are going to correspond to locations on the protein that's encoded for by DNA. And this is all mutations, and this is just the ones that were G to T, which is the typical mutation of aflatoxin and benzpyrene. So this is in mutations in lung cancer. A colleague at City of Hope, which is where I do, a medical center where I do research, Gerd Pfeiffer, in the 1990s decided to do a lab experiment to try to relate benzpyrene in cigarette smoke to P53 DNA damage. He did 2 things. First, he took normal lung cells, and he added benzpyrene to it. That's this active ingredient from cigarette smoke. And he asked the question, where does the benzpyrene bind along the DNA? And it turns out it binds at the same places where mutations happen in cancer. The same places where mutations happen in cancer. Then he biopsied the lungs of smokers and took their DNA out and tried to catch this benzpyrene in the act. Look for benzpyrene coating on the DNA in smokers' DNA. And he found, indeed, just as he found in the cells, the same place where there was cigarette smoke damage. So here is the results of that experiment, and it shows here in the top is the benzpyrene, is the experiment on the cells, and here in the bottom is the actual locations of benzpyrene on DNA in the patients. Again, it's the same place where the mutations actually happen. So these experiments had closed the loop, essentially. They had found the smoking gun. A specific carcinogen, that's benzpyrene, from tobacco, binds to a specific gene, p53, causes a specific mutation, which leads to an inactive protein, which leads to genetic damage, which leads to cancer. The result of this was that tobacco companies made settlements out of court. The state attorneys general made huge settlements in the multiple billions of dollars, and no tobacco company now argues that cigarette smoke is not connected to cancer. In this lecture, I'm going to try to describe experiments we can do to figure out if something causes cancer. We can't always do what they did with benzpyrene, right? To zero in on certain things, just because there's so many chemicals out there in the food that we eat, etc. First, I'll describe testing for carcinogens that uses model organisms and cells that can do it very rapidly. And then we'll see how these tests work. Are they good or are they bad? Secondly, I'll describe carcinogens that are natural and man-made from these tests. And finally, the concept of risk analysis. Now, there are thousands of chemicals. There's 3,500 chemicals in tobacco smoke. There are thousands of chemicals in all the foods we eat. There's thousands of synthetic chemicals we're exposed to. How do we know which ones have cancer? We really don't know, right? And the only way we can do that is to look to try to test rapidly for something that might damage DNA. So now I want to describe some tests that are rapid tests for carcinogens. Now, testing for carcinogens uses model organisms or cells because we want a quick result. We can't afford to do 30 years worth of epidemiology on thousands of molecules and then do laboratory tests and zero in on a certain gene for thousands of molecules. We just can't afford the time. So the logic is, first of all, carcinogens damage DNA. They cause mutations and DNA damage. And so the carcinogens that damage DNA could end up causing cancer as well. That's the first assumption. Something that causes mutations causes cancer. Second, simple organisms can act as stand-ins for humans. Is a rat the same as a human? Well, kind of. It's got lots of the organs that humans do, etc. Is a bacteria cell the same as a human? Well, kind of. It's got DNA and lots of things, so we can find out if there's DNA damage. But the basic mechanisms of biology are the same in all organisms. That's from evolution, really. But it's sometimes hard to interpret. And third, of course, we want these tests to be cheap and rapid. The first tests I'll describe are tests on bacteria as a model organism. Bacteria are very easy to grow in the laboratory. They can grow millions of them in a very short period of time. They form colonies of bacteria from one single one. You can count the colonies, etc. Bruce Ames, University of California at Berkeley, developed what's called the Ames test, testing for carcinogens. And what this is simply you put a chemical mutagen onto some bacteria, and you ask the question, do they grow into colonies or not? So here's potential mutagen A, and you test it on the plate. Take a look at the plate, and you might see a plate that has a lot of colonies, and you count those colonies, and you say, oh, it's causing mutations. We set it up so the more colonies there are, the more mutations there are. B might have a clear plate, might have very few colonies, so that might not be a potential mutagen. And of course, you do an untreated control in the experiment to make sure that you have no, the spontaneous mutation rate is quite low. These are easy experiments to do. I've done them with students in the teaching lab, and you get results pretty quickly. You can get them overnight. Now, you recall, chemicals get changed by the liver into something more active, so you can actually, before you apply these to the bacteria, incubate them with the liver extract and see, and then test the molecules after exposure to the liver extract. What are the results? Well, there have been a lot of results of Ames testing. They give you some indication that something damages DNA. here was one of the first. It was kind of an exciting article. It's quite famous. A carcinogen test for hair dyes, and they found that hair dyes are mutagenic. They cause mutations, some of them. They tested various hair dyes. Some of them caused mutations, some of them didn't. This was sensational because many people are exposed to hair dyes. I'm not going to tell you about myself. And the hair, especially look at people who work in beauty salons. The stylists are exposed to hair dyes a lot. So this set up a lot of epidemiology on these people. I'll show you the results shortly. So many experiments along this line were done. All sorts of things came up positive on the Ames test. People would say, well, now, come on, that's not bacteria. It's not the same as a human. I know it damages DNA, but humans can repair, all this kind of stuff. So they said, OK, let's look at human cells. That's a second type of test. We can take human cells in the laboratory dish and expose them to something that might cause cancer and see if we get cancer cells. And there are many ways that you can measure cancer cells. Another type of analysis of human cells look for DNA changes indirectly. When chromosomes are duplicated when the cell is about to divide, the two chromosomes, the two new DNA molecules lie side by side. And you can actually have different dyes to stain them. So you could have one stained a certain color and the other not stained. And normally, of course, these would remain separate, and then they'll separate during cell division, where one cell will get one of them, the other cell will get the other. But before cell division starts, we can look for changes in the chromosomes. These are called sister chromatid exchange. And when you see checkerboards such that there's been a crossover or a recombination or a breakage and reunion of the chromosomes, as we saw in the leukemia, then we can say, oh, this agent, even in the test tube, even in the laboratory dish, causes chromosomes to break and rejoin. So that's another way of looking for genetic damage. The third test I'll talk about are animal tests. Obviously, these are going to be animals we can use in a laboratory that are somewhat similar to humans. Typically, they're going to be rats and mice, rodents. So here is a cartoon that says, oh, you have a rodent in the laboratory director's desk with a suitcase checking in. And he says, and we'll give you all the carcinogens you can eat. What a deal, right? What they do is they can do two things. They can either take some cells in a laboratory dish and expose them to the possible cancer-causing agent, and then implant the cells into an animal and watch and see if a tumor grows. Or they can actually take the animal itself and expose the animal to the possible cancer-causing agent and see if the animal itself gets cancer. So that's two ways of doing it. So here's the animal testing. You have a control group that's untreated, and you have and you have experimental group that's treated, for example, with benzpyrene, that cigarette smoke thing, and you end up with tumors, etc. This is a nude mouse. It's nude because it doesn't have hair. It's a hairless mouse. But it doesn't have an immune system either. And so you can watch tumors grow in this animal. And it's a very effective way of doing cancer research because the tumors grow quite rapidly and it will actually not reject a human tumor. So these are animal experiments. These are mammals. They're similar to humans. Are they similar enough? It's hard to say, isn't it? Testing on these animals is quite expensive. And it costs between $10,000 and $20,000 because of all the animals, et cetera. It's going to take months to do. It's not an overnight test. But people will say that's more reasonable, but you need a lot of animals if we have thousands of chemicals to test. The ultimate test, of course, is to test primates, monkeys, right? This is going to take many years and millions of dollars, okay? So here's an example. This is saccharin, which is an artificial sweetener. You'll see this in stores, restaurants, etc. An experiment was done where monkeys were fed this stuff for years and then asked the question, what happens to them over time? Do they get cancer or do they not get cancer? And the reason the experiment was done was when saccharin was fed to laboratory animals, some of them got cancer. And in fact, at one point, saccharin was actually banned as an artificial sweetener. And you would see signs saying, don't have this stuff, or if you do, do it at your own risk, right? There was a food label warning on saccharin. So they fed the monkeys for a long time. And here is the result of the monkey trial. Here is the saccharin notice. It was posted for legal purposes, saying it causes cancer in laboratory animals. Here are the rhesus macaques who were fed. 20 monkeys were fed 10 times the human dose of saccharin every day. They died after 17 to 25 years of natural causes. Not one of them died of cancer. So we'd say, well, that's the ultimate. It doesn't cause cancer in laboratory animals. But think of the expense. We're going to multiply thousands of chemicals by millions of dollars. Well, let's look at the tests that are done, the rapid tests, the ones that might give us a good clue, right? And the rapid tests, you recall, were first bacteria tests, and then the human cell tests and the chromosome tests, right? The human chromosome test. Let's look and see if they're any good. Well, how are we going to evaluate those tests? Okay, now we're going to try and evaluate those rapid tests. How are we going to do it? The easy way to do it is to say, okay, I'm going to take something I know definitely causes cancer, and I'll put it in those bacteria and I'll see if they get mutations. Something that definitely, aflatoxin, something from the IARC, the International Agency for Research on Cancer, listed as a known carcinogen, you remember. Apply to the test. They all should test positive. None should test negative. On the other hand, let's take something we know doesn't cause cancer, like sugar. Okay? Let's hope that table sugar does not cause cancer. No evidence that it does. So we'll put sugar on and we'll say, does that lead to positive tests in this mutation? Does it cause mutations? If it does, then those tests aren't very good, is it? Because they're picking up things that we know don't cause cancer. Here are the results. So let's look at the data. So they tested 44 substances on the bacteria test, on the cell test, and on the chromosome test. And remember, in a perfect world, all of them should be positive. If they all cause cancer in laboratory and we know they're causing cancer, all of them should be picked up as positive. What actually happened was, not all of them, 20 out of 34, 32 out of 34, 31 out of 44. The test missed 30 or 40% of them. Now let's look at non-carcinogens, things that don't cause cancer. They tested 29 in the 3 tests. None of them, right, sugar, whatever, none of them should be positive. None of them should be causing mutations or being picked up here. And unfortunately, some of them did. Overall, these tests are about 60% good. That's not too good, is it, for informing public policy as to what we should be doing? So we're kind of in a difficult situation here. By the way, let's follow up the epidemiology on hair dyes, which came up positive in the original test. Are they carcinogenic? And the answer looks to be no. There's been extensive epidemiology done. There have been case control, cohort studies, etc. Cohort studies, case control studies. The relative risks are all around 1. So after long-term studies, it turns out that this alarm raised by the short-term testing is not really for real. So this is not I'm not trying to give you the answer. I'm just trying to tell you what the situation we're in is, which is not a good situation for short-term testing, but it's what we've got. Carcinogens are both natural and human-made. Tell me what, well, you could try to say, well, what does that mean, right? Natural and human-made. It means, well, for example, sugar, right, is natural, and sweet and low or something may not be. It's human-made, right? Many carcinogens have been suspected by population studies. This suspicion for carcinogen is made stronger by rapid testing or even stronger by epidemiology. There's a general feeling that maybe many people have is, most things that cause cancer are artificial. Food additives, artificial sweeteners, etc. But, let's consider tobacco, right? Tobacco is natural. We're burning things that their products are in the plant, and the same molecules that get into us are in the tobacco leaf anyway. Benzpyrin is there, nicotine is there, even before burning. Organisms have evolved ways to defend themselves against their enemies. So an animal can run away, right? If a bear is chasing me, I can run, right? I can run away from the bear and get away. If you're a plant, you can't run away. You have to have a mechanism in place to defend yourself against your enemies. Now, plants have enemies. Insects chew up plants, right? The larvae, the worms, the adults of insects chew up plants. Molds can land on plants, and viruses and bacteria. So lots of critters can land on plants and make their life miserable or kill them. So how are plants going to defend themselves? They use chemical defenses. What are some of the chemical defenses? I'll give you 2 examples. Nicotine is a neurotoxin. Affects the nervous system. It's made by tobacco. And here's a tobacco leaf, and on the leaf is a tobacco hornworm, a immature form of an insect that and the worm chews up tobacco leaves. It's a major pest. If the nicotine level in a plant is very high, and this experiment has actually been done, if you hype up the nicotine level high enough, you will kill all the worms because the worm, it's poisonous. It's a neurotoxin. It's poisonous to the nervous system of the animal, of the worm in this case. So it's great. The plant has things that will kill animals. Here's aflatoxin. We've seen this before. Here's the molecule. It's on a fungus. It's on peanut seeds, for example. Again, the fungus is defending itself against being eaten as well. Aflatoxin is a potent genotoxin, right? It binds to DNA. So there are hundreds of these what we call plant pesticides. We're eating them all the time. And if you start testing them in these rapid tests or even in animal testing as well, a lot of them are positive. A lot of them cause DNA damage and cause cancer in laboratory animals. Let's just look at cancer in laboratory animals. And here's a list. And you don't need to memorize this list of carcinogens. These are plant molecules that plants generally use for defense. The list is rather long. This is not all of the things on the list. And here are the plants that make these things. We'll start absinthe, allspice, anise, apple, apricot, banana, etc. Something you eat is in this list, guaranteed. So when you eat it, you're eating a carcinogen that causes cancer in laboratory animals. We're in trouble, right? But let's go back to epidemiology about risk factors for cancer. This is a pie chart I showed you earlier on in the course. Here's the pie chart for molecules and substances and cancer. You'll recall the major factors involved in cancer from epidemiology are tobacco, dealt with that, and the one we're dealing with now, diet. So how do we analyze the risks of these things? How do we analyze the risks of diet, for example, when there's so many molecules, as I just showed you, that can cause cancer? We want to know things like how much we're exposed to, how much cancer you can get from that particular exposure. Now I want to talk about something called risk analysis. This is a matter of public policy where people in government, consulting with people, with the overall population, and experts in medicine and science all get together around the table and say, how do we analyze the risk of something that we're going to be exposed to? And the risk I'm interested in this instance is the risk of getting cancer. There are 4 steps to risk analysis. The first step is hazard assessment, hazard identification. And you just have to ask the question, does this stuff cause cancer? The second is dose response assessment. How much cancer does it cause at what dose, right? At what exposure? How much aflatoxin do you need to get cancer? Of how much? Third, exposure assessment. How much are we exposed to every day? And this is a calculation. And finally, fourth, the risk characterization. Given all those facts, you're exposed to a certain activity or thing, and that thing has the following risks. What do? The risk calculation. We do risk analysis all the time. For example, get in your car. Okay? Every year, 40,000 or so people in the United States are killed in motor vehicle accidents. There are 280 million Americans, so your chance of getting killed in a motor vehicle accident if you drive for a year, just on a statistical basis, is 1 in 7,000. Now, you can do many things to minimize your risk by being a good driver, but it's still, you're doing a risk. You take a risk every time you eat food, right? Obesity, other things. But people do the risk analysis, and smokers do this all the time. They know the risks, but they're getting a benefit from it. In other words, does the benefit outweigh the risk? Let me discuss the 4 aspects now in turn. Remember, hazard analysis, dose response, exposure assessment, and risk characterization. First, hazard analysis. The way we're going to get information on things that cause cancer is from population studies, from epidemiology. We could also get it from basic biology, something we know causes mutations, DNA damage, or from these rapid tests. Even though they're not the best, they're something we've got, bacteria tests and chromosome tests and even animal tests. So we can kind of get an idea of, from rapid screens of molecules or substances, how much cancer you get. Now, how much, of course, is really the dose response, right? The dose response can be gotten on rodents, for example. You can calculate the dose, typically, what doubles the rate of cancer in laboratory animals. Here is some dose response curve and for rodents for a bunch of things. And this is for those who remember your math, this is a logarithmic scale. So we start at the bottom with 10 nanograms. A nanogram is a 1 billionth of a gram. And here's a millionth of a gram, and here's 10, 100. This is going up logarithmically on that scale. And if you look at the substance in the smallest amount is this stuff, which is dioxin, which is very potent as a cancer-causing thing in laboratory animals in teeny tiny amounts. But there's some natural things. Mitomycin C is natural. Aflatoxin is here. So you get an idea, right? You get an idea of different substances and how much will double the rate of cancer in laboratory animals. So our next step, now we know the dose response, we know that something damages DNA, causes mutation. The next step is exposure calculation, right? How much are we exposed to every day, right? How much is in the diet? How much is there, right? Well, to do this, we have to calculate how much people eat or are exposed to a substance in a given day. So we have to isolate the substance and be able to chemically detect it. And then ask the question, how much gets in? And we can make an index of exposure. Here's what I mean by the index of exposure. It's called the carcinogenic potency index. And what it simply is a ratio of the daily human exposure of something, right, in an amount of stuff per amount of body weight. So it might be if you want to do in a metric system, in milligrams of material that I'm exposed to every day that gets in my body, per kilogram of body weight. If you want to do it, ounces of salted meat versus per pound body weight per day, right? That's the dose you're exposed to. And you divide that by the dose to double cancer in laboratory animals in milligrams per kilogram per day in the same units. So what does that mean? It means that if this index is 1, it means that on a daily basis, a human is exposed to an amount of material here that will double the rate of cancer in laboratory animals at the same dose. Okay? So the higher that ratio, the worse the situation is for people. Let's look at some substances in the carcinogenic potency index. This has been done for hundreds and hundreds of substances. There are some artificial things, there are some natural things. The one that has the highest rate that's been tested so far is home air. There is formaldehyde in the insulation of almost all buildings. And when the air is inside a building, it's being recirculated in an air-conditioned building or a heated building or an unheated building, but it's being recirculated. The formaldehyde builds up, and every day it builds up to a certain level. And if you calculate that level and you look at how much causes cancer in laboratory animals, it's 40% of the level that doubles the cancer in laboratory animals. Eating an orange. Limonene is a natural molecule. It has a potency index of 0.1. Coffee. Caffeic acid. Don't show this to certain companies. Mushrooms, hydrazines, a little bit high. Now let's look down at some of the things that we worry about. DDT, before it was banned, this was the maximum permissible level. 0.002. That's 1/200th of home air. There's bacon and nitrosamines down there. Dioxin, which is highly potent. The fungicide that was banned is extremely low. What this table tells us is there are natural things that cause cancer, and there are artificial things that cause cancer. But on a dose basis, it looks like some of the natural things are pretty bad up there that we're eating all the time. And there are many more examples that I could give you of this. Related to this is the issue of low dose. The data in science can take doses down to a low level when we're testing for carcinogenesis, cancer causing, but they can't go very low. Let's think about this. Pancreatic cancer has a rate of 40,000 new cases a year, 1 person in 7,000. If you double the hits, that would mean 2 in 7,000. That's a lot of mice, right? You're going to need 7,000 mice, 14,000 mice just to get a result. So it's quite difficult to do. So now we come to low doses, and what do? Well, we could say, well, at very low doses, it may be that, Okay, this low dose, it may be that the cell repairs things, so we don't need to worry about low doses. It may be that there's a linear relationship we do need to worry. We really don't know because we can't take things down that low. The final step in risk analysis is risk characterization, right? And risk characterization means, what do we do about this? The only international environmental agreement is the Montreal Protocol, which banned Freon, chlorofluorocarbons, because they reduced the ozone hole. Now, the ozone hole was being reduced by these compounds and would let in more ultraviolet light, which causes cancer, as I indicated in the last two lectures. The risk analysis said there would be a 1.5% increase in non-melanoma skin cancer per 1% ozone decrease. The result was ban it. Okay? Ban that stuff to keep the ozone layer there. We don't want an increase in skin cancer. The state where I live in has an Environmental Quality Act that says exposure to humans to something we know causes cancer, and they're not clear about that. It's not on the IARC definite list. It could be the probable list. That increases cancer rate by 1 in a million lifetime can't be used in a new project. So a project's being built, and it says we're going to increase the cancer rate by 1 person in a million in the exposed population. Well, wait a minute. 1 person in 3 already gets cancer. So we're going from 333,000 to 333,001. Finally, there's a Delaney clause that says in the passed by the American Congress, it says the Secretary of FDA does not approve for use in food any chemical additive found to induce cancer in man in any tests at all, even and in animals as well, even if it causes cancer in laboratory animals, not just man. So there's no risk analysis here, there's no exposure, there's no dose analysis, just ban it. The extreme of this is something called the precautionary principle, which says if we can't prove that something is harmless, don't be exposed to it at all. That's the extreme of this kind of reasoning. I guess the moral of the story is that politicians need to take a science course or watch the course that I'm giving here. So carcinogens initiate transformation from a normal cell to a cancer cell. In the next lecture, we'll go into the cell and see exactly what happens. Thank you.